Hello friends and viewers. In this video I am going to talk about the innate immunity and their components and how these components are functioning in our body. From my previous videos, we now know that there are two types of immunity or immune response, the innate and the adaptive. The innate is very non-specific type and they are inborn and existing from birth and do not change over the age and do not have memory. But the adaptive immune system has very specific and change over aging and have memory. It looks like adaptive immune response or immune system is very professional to combat the disease. So why we need the innate immune system, which is very non-specific and do not have the memory? So I can give you at least two reasons why we need the innate immune system. The first one is like that the innate immune system is very rapid in response. In most of the cases, we will die if innate immunity do not come first, because the adaptive immunity takes at least 4 to 7 days to mount a good and winning immune response to the pathogen. So the innate immune response is very important for the pathogen clearance for the first week of the disease. And second cause is scientists have shown that some deficiency of the innate immune system, like the IRAC or interleukin 1 receptor kinase 4, there are certain type of bacteria is increased in our body, like the gram-positive bacteria, and also the deficiency of the NK cell, which is considered the component of the innate immunity, increase the herpes virus infection in our body. And also the innate immune response make some linkage to the adaptive immune system to mount the higher and very specific adaptive immune response. So in total we need the innate immune system. So next we will see that what are the main components of the innate immune system and how they are actually functioning in our body. To make it easy, I have categorized these innate immune components in different category, like the barrier category and the cell-mediated effector cell category and the humoral category. In this category also I have categorized the cytokines and the effector proteins separately. And there are some process like the phagocytosis, cytotoxicity and inflammation. And there are some receptor like the pattern recognition receptor. They are divided into toll-like receptor, which are also the extracellular and the intracellular, and nucleotide binding oligomerization domain-like receptor or knot-like receptor, and Rick-like helicase. Those all are the receptor on the cell, and the NK cell receptor. So with this component, the innate immune system mount a immune response in the first week of the bacteria or virus entry inside of our body. So next I will describe each of these component separately and try to make you understand that how they are actually functioning in our body. So the barrier can be divided into the physical and the chemical barrier. So the first one is the skin. So this is the human body. So skin is the first line of barrier. Because of this skin, any bacteria or virus cannot enter inside of our body. And then we have the epithelial layer. Though we think that our lung and the intestine is inside of our body, they are mainly the exposed part of our body, because through the air and the breathing, most of the bacteria and the virus enter inside of our lung. For example, every year we are infected with the influenza and we have the running nose and various problems in our body. And by eating the foods, we get infected with lots of bacteria and the virus through the intestine. Sometimes we are infected by the parasites and the bacteria that can causes the bowel pain, and we have pain inside of our belly. This can be infected by the oral consumption of the bacteria or parasite with our foods. So in that region and also the reproductive tract, we have a lots of epithelial layer that help to prevent of these bacteria and the virus to get inside of our body. Next, the mucous membrane. Mucous membrane also the layer of the epithelial cell, and one or more layers of the epithelial cell makes this mucous membrane or mucosa, which surround the different cavities of the body like the intestine and the lung and different parts of the body. So this mucous membrane and the epithelial layer are almost similar pattern. They prevents the entry the virus of the bacteria inside of the body. And next, the hydrochloric acid, which is specifically produced by the stomach of our body, because when we eat something, those food have the live bacteria or the virus. When we eat this bacteria and virus with the food, they directly go to the stomach, and stomach produce the hydrochloric acid, which pH like 1.5 to 3.5. In this very lower pH, which is very acidic, the food bacteria or virus are chemically killed by this hydrochloric acid. So this is a type of chemical barrier. And the mucous membrane and the epithelial layer, they have a specific type of cell that can produce the sticky secretion of this epithelial barrier. And with this sticky secretion, most of the bacteria and virus, they are stuck to this epithelial barrier, and they cannot get entry inside of our body, and also they cannot replicate themselves, and they cannot attach to the epithelial cell to get entry inside of our body. In this way, they are actually physical barrier. Next, the defensins. The defensins are very small peptide. Normally they are 18 to 45 amino acid peptides, and they are cationic peptides. The cationic peptides are termed because the total number of charges of this peptide are positive. At least 2 to 3 total or net positive charges are carried by this peptide. And these positive charges are help in killing the virus or bacterial cell. And these defensins or the peptide are called the host defense peptide. They are produced mainly by the epithelial cell and the neutrophil and the granulocyte, and they attack just after entry through the intestine and through the lung, and they are called the first line of defense because when the bacteria or virus get inside of body, they are secreted first from the epithelial cell. The most mentionable defensin or the host defense peptide are the alpha defensins and the beta defensins. Next is the lysozyme, which is secreted by the tears and the human saliva, and this lysozyme cleaves the peptidoglycan on the bacterial cell surface. Because the bacteria have the peptidoglycan on their cell surface, which is very specific for the bacterial cell wall, not the human cell. So this lysozyme can specifically cut those cell wall, and in this way this lysozyme help to break down the bacterial cell wall and gives the protection from those bacteria when they enter inside of our body. Next are the cell-mediated effector cells. In this category, we have different types of cells like the neutrophil, macrophage, and the NK cells, and innate lymphoid cells, platelets, mast cells, and the intraepithelial lymphocytes, also the dendritic cell. So when a bacteria or virus get inside of our body, the first cell who is attack this bacteria or virus is the neutrophil because they are very high in number in blood. So first this neutrophil come in contact to this bacteria and they phagocytosized those bacteria and virus. And the phagocytosis is a process through which this special type of cell engulf the bacteria in a whole or parts of its components. And here I am going to show the simplest way of how this phagocytosis actually happen. So these are the phagocytic cell, the neutrophil or the macrophage. They have the extra projection of the cell surface, and these are the bacteria and the virus. Bacteria and the virus, they have a special type of cell surface molecule that can be recognized by this neutrophil and the macrophage. And when this phagocytic cell recognize that this is the bacteria and not the self cell, they actually attach this bacteria to their cell surface and then they engulf. That means the bacteria get inside of this phagocytic cell, the neutrophil and the macrophage. The process is called the endocytosis. This can be also the receptor mediated and non-receptor mediated. So the receptor mediated and the non-receptor mediated. And in this way the bacteria go inside of cell, and inside of cell there are a different mechanism through which these bacteria are chopped down with different type of the enzymes, and through this process those bacteria are killed inside of this phagocytic cell. And neutrophil and the macrophage, these two type of cell have the capacity of phagocytosis, which are the innate part of the cells. And after the neutrophil, the macrophage come from the blood. In the blood, the macrophage termed the monocyte, and when they come from the blood to the tissue to see and kill the bacteria or virus, they are termed the macrophage. And the macrophage is termed with different names with their specific tissue. Like when they go to the lung, they are called the alveolar macrophage, and when they go to the liver, they are termed the Kupffer cells. But they are almost the same type of cell which come from the monocyte of the blood. So this type of neutrophil and macrophage cell come in contact to this bacteria and virus and then try to kill those bacteria and virus by the process of phagocytosis. Sometimes we found some pus cell inside of our body. These pus cells are produced by the dead cell of the neutrophil, because the after action of the neutrophilic phagocytosis and killing the bacteria, those neutrophils also die. So they are deposited as a white pus cell inside of our body. Next, the NK cell. NK cell are called the natural killer cell because this cell can recognize which cell of our body have the MHC molecule or not. So depending on the MHC molecule, they can detect that our cell is infected or not. And this type of cell especially very important for the virally infected cell and the tumor cell. When our body cell are infected by the virus or they alter the cellular mechanism and turn into the tumor cell, they loses the capacity to express the MHC1 molecule. But in our body, every cell produces the MHC molecule. So when this NK cell see that our one body cell have the missing the MHC1 molecule, then this NK cell can trigger on, and then they kill those type of cell who do not have the MHC molecule. And the process through which this NK cell kill the cell is called the cytotoxicity. In the similar fashion, the adaptive immune system, like the CD8 T cell, kill the virally infected cell. But those process is mediated by the other mechanism, but not by like the NK cell and the MHC1 missing concept. And also, since this NK cell can detect the MHC missing cell, they can act very rapidly, and it has been shown that they can also attack and mount a good response at the very early of the immune response, like the 3 days after the infection. In contrast, the CD8 T cell, which can take almost 7 days to come into action. So here is the simplest way of the how cytotoxicity can kill the infected cell. These are the NK cell, and this is our virally infected cell, or the turned to tumor cell inside of our body. They do not have the MHC1 molecule. So this NK cell can detect through their receptor that these cells are missing of the MHC1 molecule. Then these NK cells secrete some chemical nearby of this infected cell, and through this chemical, which are also the enzymes, they get inside of this infected cell, and they chop down their DNA and all of the protein, and through this process they kill the whole of the infected cell, and the process is called the cytotoxicity. The similar fashion of this cytotoxicity is happened for the CD8 T cell, but that is for the adaptive immune response. Next, the innate lymphoid cell. The innate lymphoid cell are almost similar type of the lymphoid cell, but they do not have the CD3 molecule. So they are termed the innate lymphoid cell, and they almost same categorized by the Th1, Th2 and Th17 type of cell. And here we categorize the innate lymphoid type 1, type 2 and the type 3 type of cell. I have another video where I have also described these 3 type of cells separate, so you can see that video for more clarification. Next, the platelets. Platelets, though they are mostly involved in the other function of the body, but they are also categorized as a part of the innate immune system, because when the blood clotting happens, these platelets help to prevent the release of the immune cell from the blood to the outside of the body. And also the platelet have been shown to produce a lot of cytokines that help in the immune system. Next is the mast cell. Mast cell are mainly the tissue resident cell, and they produce mostly the inflammatory cytokines and the molecule that help to guide the other cell to come into the site of infection. This mast cell produce some chemical like the histamine and the other chemicals, which help to increase the blood flow and the permeability of the cell that is associated with the blood vessel, which actually facilitate the immune cell to come through this blood vessel. And the process is called the extravasation or the diabetes. And the inflammation, which is a big process in the immune system, is also associated with this mast cell and other types of innate immune cells, where there are many cytokines and the immune modulator, which are the protein or peptides, are released into the tissue, and through this release of the molecule, the whole system alert the immune cells to come in the targeted region where the bacteria or virus get inside of the body. Then comes the intraepithelial lymphocyte. They reside in the epithelial barrier, especially the inside of two epithelial cells. That's why they call the intraepithelial lymphocyte. They are like the lymphocyte or T cell, but they do not need the activation like the T lymphocyte. They can instantly release the cytokine and kill the virus or bacteria very quickly. So they are very important in the innate immune response. And lastly, the dendritic cell, which is called the bridging cell between the innate and the adaptive immune response. They are like the phagocytic cell, but in most of the case they work not only for the killing of the bacteria or the virus, but they try to engulf those bacteria and the virally infected cell, and they chop down the viral or bacterial proteins into peptides, and then they present to the T cell. They express those peptides with the MHC molecule, like the MHC1 or MHC2, on their cell surface. And by expressing this peptide with the cell surface MHC class 1 or MHC class 2, they help to direct the T cell to induce the immune response. That's why they are called the bridging cell between the innate and the adaptive immune response. And next, the pattern recognition receptor. These receptors are located on the cell surface of the neutrophil, macrophage, and this type of phagocytic cells, and they help to recognize the pathogen associated molecular pattern, which are also some molecules that are expressed on the bacterial surface. Those molecules are categorized into different types, like the most popular one is the lipopolysaccharide or the LPS, and also the peptidoglycan and the flagellin are the pathogen associated molecular pattern, which are recognized by this pattern recognition receptor on the cell surface of the phagocytic cell. There are at least 3 types of this pattern recognition receptor. Those are toll-like receptor, node-like receptor, and Rick-like helicase. The toll-like receptor are categorized in the extracellular and the intracellular. And at least 13 types of murine TLR and the 10 types of human TLR are discovered nowadays. And TLR1 and TLR2 TLR 4, 5 and 6, those TLR are extracellular, and the TLR 3, TLR 7, 8 and 9, those TLR are intracellular. They reside inside of the endosome. And these TLR 1 and TLR 2 recognize the bacterial lipopeptide. TLR 2 can recognize the bacterial peptidoglycan. TLR 4 can recognize the bacterial lipopolysaccharide or the LPS. TLR 5 can recognize the bacterial flagellin, which is the protein of the bacterial tail. And the TLR 2 and the TLR 6 combinedly can recognize the bacterial lipopeptides. And the TLR 3 can recognize the double-stranded RNA, which are mainly coming from the viral DNA. And the single-stranded RNA is recognized by the TLR 7. TLR 8 can recognize the single-stranded RNA, and TLR 9 can recognize the CpG-DNA. So through this different type of TLR, different types of the bacterial and viral components are recognized, and then this phagocytic cell give rise the inflammatory response, which can trigger the attraction of the other immune cells. The knot-like receptor, they are called the nucleotide binding oligomerization domain-like receptor, or in short the knot-like receptor or the NLR. They are intracellular sensor of the pathogen associated molecular pattern. Those are come from the bacterial surface. For example, the bacterial peptidoglycan, and these knot-like receptor are expressed on the cytosol of the phagocytic cell like the neutrophil and the macrophage. In this knot-like receptor protein, there are at least two types of domain. Those are found in the N-terminal of the protein. The first one is the caspase-rich root domain, which is called the card, and the pyrene domain. Mammalian knot 1 and knot 2 are very well recognized in the knot-like receptor family, and these knot 1 and knot 2 have the card domain. This card domain help to signal through the intracellular pathway and produce the NF-kappa B, which is finally produce the inflammatory response. But the other knot-like receptor family, like the NALP family, which is also called the knot-like receptor proteins, they have the pyrene domain, and they are the component of the inflammasome. Inflammasome is a big, large multimeric protein which is associated with the production of the inflammation. And this multimeric protein, with the help of knot-like receptor, they produce the interleukin 1 beta and interleukin 18, which by turn produce the inflammation, and they call for the help of different lymphocytes in the blood circulation. Then the RIC-like helicase. RIC1 is actually the retinoic acid inducible gene 1. So they are called the RIC-like helicases. These RIC-like helicases are very essential to viral recognition in the cytoplasm of this neutrophil and the macrophage and the innate phagocytic cells. They can recognize the viral double-stranded RNA. And these RIC-like helicase are comprises of at least 3 types of proteins. The first type is the RIC1, second type is the melanoma differentiation associated gene 5 or MDA5, and the third one is LGP2, the first and the second, which is RIC1 and the MDA5. These two proteins have the N-terminal CART domain and the C-terminal helicase domain, but the LGP2 do not have the CART domain. So if you remember that if a protein have the CART domain, they have the ability to induce the NF-kappa B pathway. So these two first protein can lead to the NF-kappa B pathway, whereas the LGP2 cannot. The helicase domain of the RIC-like helicase show very high similarity to those of the dicer protein. The dicer proteins are a type of nucleases that very essential for the generation of a small interference RNA. Next, the NK cell receptor. The NK cell receptor are divided into 2 categories, which are the first one is activating receptor and the second one is the inhibitory receptor. The activating receptor is the Li49H and the NKG2D. And in this NKG2D receptor, which is like the stress induced and the MHC1 like molecule. And the inhibitory receptor, the Li49A and the P58KIR, these two are very important. Li49A receptor is similar to the mice MHC-DD molecule, and the P58KIR is related to the HLA-C molecule. Next, the humoral part of the innate immune system. In this humoral part, there are 2 categories, the cytokine and the effector proteins. In the cytokine categories, there are lots of cytokines are involved in the innate immune system, but for simplicity, I am only talking about some of those cytokines, especially which are very important. These 4 cytokines, interleukin 12, interferon gamma, tumor necrosis factor, and the interleukin 1 are very important. When an infectious agent like the bacteria come into contact with this macrophage and the dendritic cell, this cell produce the interleukin 12, which act on the natural killer cell, and by turn this natural killer cell or the NK cell produce the interferon gamma. So in this way, the phagocytic cell induce the natural killer cell, and those natural killer cell kill the infected cell, and this natural killer cell produce the interferon gamma, which induce the more phagocytosis process of the macrophage and the dendritic cell. And then this phagocytic cell produce the TNF and the interleukin 1 and some other chemokines. Those are very helpful for the production of the inflammation, because this type of inflammation by this cytokine help for the recruit of the neutrophil and other lymphocyte from the blood vessel to the site of infection. Next, the complement, which are in the category of effector protein. There are several, at least 9 complement protein inside of our body. They are circulating in the plasma of our body, and they are produced from the liver. In the normal condition, they are not too much activated, or they are activated at the basal level. But when the bacteria come into contact of this complement protein, or any bacterial component come into contact of this complement protein, then this complement protein go through a activation by 3 different cascades. I'm not going through these 3 different cascades in detail, but just for simplicity, these 3 types of cascade are termed the classical, alternate, and the lectin pathway. The classical pathway is activated by the antigen antibody complex bound to the bacteria, and the alternate pathway is activated by spontaneous cleavage of the complement 3, which is spontaneously cleaving at the basal level in our body, and the lectin pathway is activated by the mannose binding lectin. Lectin are the protein that can bind to the carbohydrate of the bacterial cell surface and can activate the complement system. So in these 3 pathways, the complement protein are activated, and finally all of these complement protein make a hole on the surface of the bacteria. By forming this hole, the bacteria get lysis and get killed. So in this way, the complement pathway kill the bacteria. But same procedure cannot kill the host or the human cell, because they have several protective proteins and mechanisms that can remove of this type of lysis and the whole formation inside of our body. And these proteins are termed the complement proteins, and the system is called the complement system, because when the antibody opsonize the bacteria, opsonization is a process of the wrapping of the bacteria over the cell surface, as if the immune cell can recognize that this is the foreign bacteria or the cell. So when this antibody opsonize the bacteria, this complement protein also bind to the bacterial cell surface and complement or enhance the ability of the recognition by the immune cells. So they are called the complement proteins. Then the C-reactive protein. C-reactive protein are produced from the liver and circulate through the blood. They are the first pattern recognition receptor that was identified. This C-reactive protein is produced in response to the interleukin 6 that is produced from the macrophage. This C-reactive protein is always in the laboratory test. This indicates that inside of body the inflammation is persisting, and in the immunology they opsonize the microbes and they also activate the complement cascade, and in this way they kill the bacteria. Sometimes the coagulation factors or the coagulation protein are also considered the component of the innate immunity, because the fibrinogen or the fibrin plays an important role in controlling the infection, and also the cellular injury by the bacteria and the pathogen initiate the coagulation and the inflammation simultaneously. And the complement activation contributes to the coagulation. So for this reason, the coagulation factors are also considered the innate immune system, and they also help to reduce the infection by the pathogen. So this was the total component and the function of the innate immune system in this video. Hope you have learned something from this video, that how the innate immune system actually work inside of our body. Thank you for watching. If you think that this video is very helpful to learn the innate immune system, please like us and also do not forget to share it.